So now I welcome all the participants, attendees, and speaker, Dr. Agam Prasad Singh, sir. As you all know, this is a free webinar series. Main purpose is to disseminate knowledge, especially in biosciences stream and allied related subject, which will help the bioscience subject. I request all the participants, attendees to attend complete lecture and fill the feedback when we share this feedback link in the chat box. Don't ask me feedback after one day or before the lecture completion. If you will fill that feedback form, so you will receive e-certificate also. Sometimes some research scholar need certificate. And this is interactive session, so you can unmute and speak, but preferably we start question answer session after finishing completion of the lecture. But meantime also, if you find that your question is very pertinent and you are not going forward without answering that question, so you can unmute and speak. No one is in mute mode, you can interact. So this interactive session, but purpose is to disseminate knowledge in different domains. And in this series, I'm grateful to Dr. Agam Prasad sir for accepting our invitation. We have already circulated his introduction in different groups, but for those who have joined now through other means, other way, through direct link. So sir is a scientist at NII. As you all know that NII is DBT Institute, and he's PhD from JNU and then spent several years as a postdoc in different institutions at USA. He has several patents, 7 patents. He has given PhD, means guided PhD of several students. He has also more than 40 good research paper in very, you can say, reputed journal, high impact journal, including Cell also. So we are grateful for his acceptance, and so I request Agam Prasad sir for this with this brief introduction to start his lecture. And again, I'm thankful to Agam sir for accepting our invitation. We are pursuing him since several months. As you all know, all scientists are busy. But this is always, I speak that this is daring work to deliver lecture online, because most of the time speakers are asking what type of audience we have. But we don't know which type of audience we have. We just share the topic, and those who are interested in that topic, they can attend. You can interact, and we have not limited other undergraduate student or postgraduate student or faculties, because our experience is sometimes graduate students are better than sometimes faculties who are teaching since 30 years, but they are teaching only classical things. That's why we are not limiting, and we are not limiting कि सिर्फ student हो या teacher हो या है ना, just we are disclosing the topic and the expert. So sir, now
Thank you,
platform is
thank you,
yours.
thank
Yes.
you, Gopalji. What I will do is I will try to make things crisp and also to a level where a master level or PhD students or people who are beyond that, they can understand things easily. Okay, and I'm also open if somebody wants to ask a question in between. I'm open to that. I have no problem with that. So, with that, can I now start my lecture?
Yes,
Okay,
sir.
thank
As
you,
I
thank
can
you.
see, there several HODs have also joined. Prabhakar ji has joined. He is in Sikkim, Nagaland University HOD. Sasikant Komu sir, his principal in here in Gandhinagar. There are several faculties also attended. Ritesh Arya is scientist in AIMS.
Okay, no problem. Whatever is the level, I'm trying to make it more generic.
Yes, sir.
So,
Yes, sir.
if there are any advanced level things somebody wants to ask, can ask me question. And also, not only during this discussion, you can give my email address to every audience, and
Yes.
they can ask my question later on also.
Yes, sir. Yes,
Okay,
sir.
so with that, I will start.
Yes. fine.
Okay, so thank you all for joining, and I'm going to talk about the host modulation or changes in the host that a pathogen and parasite makes for its survival and also to defend itself from the attack from the host side. So attack could be of multiple types. It could be from a immune system. It could be a host defense mechanism like apoptosis or other mechanism, autophagy and other. So
Mother nature, yes.
okay, so the parasite or the parasitic lifestyle is a type of non-mutual relationship between organism of different species where one organism, the parasite, benefits at the expense of the other, the host. And the what is host manipulation? Any alteration in host phenotype induced by a parasite that has a fitness benefit for the parasite. Fitness benefit means here either a growth benefit or number of progeny that it produces. So with that, I will move on to the next clip. So some of the examples I am showing here, the what type of parasites that you might be knowing. The protozoal parasite, malaria parasite, different species of malaria, leishmania, trypanosoma, and then helminths. We have different types of flukes and tapeworms, roundworms, and some of the pictures here I am showing like fleas, helminths, the mosquitoes, giardia and babesia. These are some of the examples that I'm showing. Some of them you might be knowing, and if you don't know, you can just refer back to them if you are interested in more detail. So I'm giving you here an example of a malaria parasite which infects in the liver of the host. Say for example human host. So the section on the top, the section on the top left side A is showing in red a healthy parasite, and in B you don't see any red staining, and only you see these black dots which are basically neutrophils. And in the bottom you see a red parasite surrounded by a green structure which is the parasitophorus vacuole membrane, and in D you see only green structure which is the remnant of the parasitophorus vacuole, but no red staining, but that means there is a no live parasite. So these are the images from the same sections of the infected host. So what it tells is that in A and C what you see is a healthy parasite growing, and B and D it is showing that the host is able to kill the parasite. So the conclusion is that if the parasite is not able to manipulate the host pathway, the parasite will be eliminated by the host. So this is the basic idea before we go and try to understand why host manipulation is important. So the topics that I am covering in this lecture is export of effector proteins, targeting the host transcription factors, targeting the host cell death, targeting the host autophagy, and subversion of the host organelles and pathways. These are the 5 topics that I am going to cover. So some of the references for further reading, uh, you can see this article from Pathogens, Proteomics for the Host Pathogen Interaction. This review in phytopathology discusses various methods used for understanding the plant pathogen interactions and effectors. And this third host cell microbe article, this talks about the pooled CRISPR screening with a dual host pathogen single cell transcriptome readout. This will tell you both about the host and the parasite transcript fluorescent protein or a luciferase or beta galactosidase or red protein RFP to study the localization and other details of the related genes which are suspected to be exported proteins. Then you can also do the transient expression systems to study the localization and interaction of the proteins involved as a effector function. The other methods which are used are yeast or bacterial to hybrid assays. So you can, if you want to study the protein-protein interaction, you can use the two hybrid system. And also those who don't have the wet lab resources, they can start with the pathogen host interaction database where a lot of these interaction, how they happen and what kind of proteins are involved, that can be previously learned from this database before you start your work in the wet lab. Then biochemical and proteomic tools can be used, like affinity purification followed by mass spectrometry. So the suspected protein can be purified and then used for purification of interacting partner, which can be studied by mass spectrometry. The other methods that can be used is called proximity labeling, where one protein is labeled with the one arm of the tag, and other uh, protein which is interacting, if they come in close proximity, they get labeled with another tag, and that can be studied. That is called proximity labeling. Protein microarrays can be used to study the interaction. You have one protein on the array and other protein in solution. If they interact, they will bind and you can study them. The chemical cross-linking method can be also used, where you take the one protein which may be present on the surface or purified protein, add another lysate of interacting protein, and then you can chemically cross-link them, purify them, and then do mass spectrometry to understand what kind of proteins are interacting and uh, their type of interaction that might give you the desired effect. Then surface plasma resonance or SPR. Then computational and bioinformatics tools are also employed. For example, effector prediction pipeline. Here, if you don't know about a protein or you want to know whether this could be a potential effector molecule, then you can use the prediction pipelines. Comparative and functional genomics. If you already know a effector molecule, then you could look for the similar molecule in other species or other organism by doing comparative and functional genomics. You can also use the transcriptonic RNA sequencing, where you can take the infected pathogen infected host and do a dual transcriptome of both host and pathogen to understand what kind of gene expression changes are happening, and that you can later on link it to the interaction. Proteomics similarly can be used, and structural prediction can also be used for doing the protein-protein interactions of these effector molecules. Finally, you can use the imaging and microscopic tool. You can label the fluorescent protein and do a confocal to see their localization and the interactions. You can do intravital microscopy in vivo technique. You can use bioluminescence based localization and interaction studies. And if you want to do a more detailed study at a molecular level, you can use the electron microscopy to see their localization, interaction, and other activities. Human malaria Plasmodium falciparum, there are more than 400 putative secreted proteins which are secreted outside the pathogen in the host to modulate the host. And many of these proteins come from a family of protein like PFEMP1, ripin, and stewar, and they are multiple in number. Those numbers are basically associated with uh, tackling with the immune evasion strategies of the parasite. So what happens in case of a set of malaria exported protein? Not all proteins are uh, exported in the same mechanism. Some of them are using a specific method called Plasmodium export element or Pexel motif containing protein. There are other set of proteins which are yet non-known, not known how they are exported, but nonetheless they are exported. So I will give you example of those proteins where there is a export motif called Pexel, which is if it is there, the protein will be going across the parasitophorus vacuole of the pathogen. So these proteins have a pentameric motif which is called RX, LX, E oblique Q. So if any protein have similar kind of amino acid sequence in the N terminus, they are more likely to be exported across the parasite vacuolous membrane. So this if you have this RX, LX, E motif or pixel motif. They are exported by a machinery called translocan. This translocan is made up of the protein called EXP2, then PTEX 150, hit shock protein 101. This basically helps in unfolding of the protein to pass through the narrow pore which is generated by the EXP2. So this is a specific for the malaria parasite, and malaria parasite introduces all this machinery into their parasitophorus vacuole membrane for exporting a set of the proteins into the host. So how this pixel motif was identified, as I discussed you earlier, microscopy can be used to identify the effector molecules. So what you see in the top A, uh, there are different proteins which are either within the parasite or outside the parasite. So in dark green, what you see in A is GFP tag protein present only in the within the parasite, uh, uh, within the parasite. But in B and C, what you see is that dark green, and then you see some light green in the red blood cell cytosol. So the what is the light green that you see in red blood cytosol are those proteins which are exported in the RBC cytosol. And using the computational method, you can see in this B picture that there is a R, R which is conserved. Then third position you have L which is conserved, and at fifth position you have EQ amino acid which are conserved. And in terms of on the right hand side you see the relative importance of each amino acid, R, L, E. So R and L is the most important. E can be E and Q are preferred, but it can be also taken care by the other amino acids. So many of these malaria proteins which are exported, you can see in this blue diagram that they have a signal sequence, as well as majority of them have the pexel motif. And where these proteins are involved and what kind of functions they do, if you see in the bottom, there are three different genes which has been knocked out and their function has been identified, and based on that they were further uh, further studies were done to identify their effector functions. So in the first, which is the CS2 is written, that shows a small dot like structure on the surface of infected red blood cell. These structures are called as the knobs. These knobs are required for adhesion of these infected RBC to the epithelial cell in the blood circulation. And the PF10381 and PFD1170C, these were the two protein containing pexel motif. When they were mutated or knocked out, you can see these now RBCs are swollen. So these proteins are involved in the structural integrity of the RBC. So, so these proteins are exported by the parasite into the RBC cytosol for maintaining their knob-like structure or their structural integrity. Similarly, if we take the example of the toxoplasma, another protozoan parasite, so they also have similar kind of protein export mechanism like plasmodium. They also have various proteins which they can introduce in the host cytosol, and some of these proteins which are introduced in the host cytosol, they also contains a nuclear localization signal, and therefore these proteins can also go into the nucleus of the host. You can see here some example of the protein which are entering into the host nucleus like ROP16, protein phosphatase 2C, GRA24, GRA16, and there are many more. I'm giving you just a few example. And these can be again identified through microscopy. You can see here the GRA16 label in red is present in the nucleus of the host, which is marked as a N in the picture. Similarly, GRA24 in green, you can see it is present in the nucleus of the host labeled as the N. And in dark black, you see those structure which are basically parasites, toxoplasma. So toxoplasma and plasmodium both are able to introduce the effectors in the host for modulating their function. We will talk about the what kind of functions they modulate a little later. So just like a pexel motif in Plasmodium, later on the toxoplasma proteins were also discovered that they also have a pexel-like motif, not exact but similar to the pexel-like motif. And two proteins, GRA16 wild type and GRA16, the motif where it was mutated with alanines. On the top you see the GRA16 with the white arrow, it is present in the nucleus of the host, but in the bottom if you see the second picture in the bottom where it was motif was mutated with alanine amino acids, you don't see the green protein in the nucleus, showing that this motif is important for export of this protein across the toxoplasma and to enter into the host nucleus. So this, because this is a plasmodium a pexel-like motif, so it was named as a texel for toxoplasma pexel-like motif, and typically it contains RRL X motif. The this motif both in plasmodium and toxoplasma is cleaved by a protease after the L or leucine, and the remaining protein is then identified by the translocon for the export. In the B picture you see the wild type where you see two bands in the western showing that the protein is cleaved by the protease, but on the right hand side where you have the mutated protein you see a higher side band but nothing in the lower sides, telling you that it is not cleavable by the protease. And in terms of
But
activity,
why there is less amount of protein? Because sometimes if protein amount is more, then also you see that lower band.
no,
In your
in this picture it is not shown. It is a publication from some other group, but
okay,
even if you have more amount, it will not. So that is why you see this next paragraph, which is next graph, which is showing that if you mutate, the activity is lost. So it is not about the quantity. Although I would agree with you that they should have loaded more, but this is just to
yes.
tell you that this is the thing that
Because
happens.
if cleaved amount is less, then you can see the proportionately that will be not visible.
Yeah, I mean that is the problem with the authors who did this experiment. But
Yes.
basically, this has been any other proteins, so there is no point of questioning. This is just one example. So there are multiple proteins where this has been proven. Present, it's just slightly bigger than the bacteria. There also you can see this dark green is the parasite and light green is the exported protein inside the red blood cell cytosol. And when these proteins were tagged with the MIG tag and then they were separated and analyzed in a western analysis, you can see these proteins are present in the red cell cytosol, and by doing electron microscopy they have been identified to be present in organelles like micronemes and rhoptries. So small dots, small black dots are micronemes, and the bigger dark uh, size organ is called rhoptry. So they are present in the micronemes and rhoptries of the uh, protozoal parasite like Plasmodium, Toxoplasma, and Babesia. So now I have shown you the evidence how the pathogens are exporting their proteins for various purposes. Now we will see one by one what kind of functions these proteins do. So we will take example of the protein which is present in the plasmodium. It is called the circumsporozoite protein or CS protein, and it is present in the sporozoite which is infective stage of the plasmodium. The sporozoite infect the hepatocyte inside the liver. So this protein has a multiple domains: the N-terminal domain, the middle domain that contains repeat, and C-terminal domain, which contains the thrombospondin uh, thrombospondin conserved domain, and in the end it has a GPI anchor. So in the N terminus, the labeled is P1, P2. These are basically two pexel motifs, and in the C terminus, the labeled as the TSP domain, that contains a nuclear localization signal for this protein to enter into the nucleus. So the species we have shown here, like two mouse species, Yolii bergii, Falciparum human species, Raconoid chimpanzee, and Gallinaceum is a bird malaria. So everywhere you see the Pexel 1 and Pexel 2 marked in the red color amino acid. So the amino acid location and type of amino acids are more or less conserved. So this is a essential in some of the Plasmodium species, but there are other category of Plasmodium species which contains only one pexel motif which is conserved, which is pexel 2 motif. In some of them, pexel 1 is not conserved. Nonetheless, these are important motifs and play role. So in an experiment, as I told you, the thrombospondin domain contains the nuclear localization signal. It also contains a nuclear export signal. So if you take a small region from this thrombospondin, a 9 amino acid region, and you fuse this 9 amino acid region with a cytoplasmic protein, for example, aldolase, in the bottom C and D, you see the aldolase which is present inside the cytoplasm only, and A and B, you see if it is fused with the nuclear, this NLS of CSP, or majority of the protein goes into the nucleus. Red staining tells you the boundary of the nucleus. So you can see majority of the protein is present inside the nucleus after fusion with this NLS of CSP. So that proves that these proteins has an active NLS, and they can take protein inside the nucleus. So what is the function of this protein? This protein, if you remove a small portion from the N terminus near the pexel motif, then it becomes active protein, functional protein. So that protein was labeled as the delta CS or deleted CS, has a enhanced parasite growth if you have the delta CS. If you remove the NLS of this delta CS, the enhancement is reduced. And if you add another active NLS, like for example SRY NLS from another protein, it can again regain the function. And if you mutate this positively charged amino acid, uh, 3R, 3T means 3 R's has been mutated by the 3 threonines. So this loses the function. What is the implication of this protein is having this kind of NLS, is that this protein uses importin alpha 3 and 4 to enter into the nucleus. So you can see here it binds to importin alpha 3 and alpha 4, and to some extent it also binds to alpha 6. So the another protein which is involved which is involved in the entering of the nuclear factor kappa B, which is a immune regulation related transcription factor, uses the same importin alpha 3 and alpha 4 to enter into the nucleus. So what parasite is doing is that it is introducing the CSP protein into the host cytoplasm, and it is entering into the nucleus. So by their sheer number, they overpower the activated NF kappa B, which is smaller in number. So thereby they don't allow the activated NF kappa B to recruit the proteins or activate the promoters under the control of NF kappa B. Therefore, they regulate the immune action of the host cell. So this was proven in an experiment where you see in the normal HeLa cell, the amount of NF kappa B in dark color you see present inside the nucleus. And if you overexpress this delta CS protein in the same cell line, you see a very less amount of NF kappa B going into the nucleus. And by transcriptome analysis, it was identified that more than 40 genes under the control of NF kappa B was down regulated. Some of the genes which are down regulated are listed here in the yellow shaded area. You can see the immune related genes like CCL20, INHB, Inolase 2, PTGS1, they are all involved in the different function of the immune system. They were all down regulated when the delta CS was overexpressed. This shows that the parasite is exporting actively this protein to manipulate the host immune related function. Similarly, toxoplasma also introduces a protein called ROP16, which also moves into the nucleus. You can see here in the bottom, dark green and the nucleus in the ROP16 type 1. And in the third lane, you see, so if you mutate the NLS of ROP16, you don't see anything inside the nucleus. Why these proteins are important? If you can see the different type of uh, strains which are available for toxoplasma, they are called as a type 1, type 2, type 3 strain. So type 1 and type 2 strain, they basically control the they basically control the amount of interleukin 12 produced by the host cell. But type 2 cannot regulate the IL-2 production by the host cell. So this has a implication for the how these parasites grow inside the host cell. And here also, if you mutate the NLS of this ROP16, you basically lose the function of controlling the IL-12 production or control of the IL-12 by the parasite proteins. So what is happening here, if you see in this A picture, which is a Western blot, type 1 and type 3 are able to allow the phosphorylation of phosphostat 3, but type 2 is not able to allow the phosphorylation of stat 3. The total stat 3 is also different in case of type 1 and type 2 and type 3. So basically in type 2 there is a less induction of stat 3 and less phosphorylation of stat 3. Stat 3 has a function in regulating the interleukin 12, therefore it has a implication for the growth of the parasite. In the B panel, you see in the red you have the phosphostat, and in the green you see the protein from the parasite, and you can see phosphostat clearly accumulating inside the nucleus of type 1 and type 3 strains, but not in type 2 strain. So what are the different things that happen in type 1 parasites? Type 1 parasites, they have decreased TNF alpha, decreased IL-6, decreased IL-10, enhancement of type 3 stat 3, and down regulation of NF kappa B. This leads to unchecked proliferation of the parasite and leads to death of the host. In the type 2 parasite, the NF kappa B is up, stat 3 is down, IL-6 is up, IL-23 is up. Although these parasites proliferate fast, but they are not able to kill the host. In type 3 parasite, the STAT3 is up, NF kappa B is down, TNF alpha is down, IL-6 is down, IL-10 is down. These parasites, they grow slowly and also they lead to chronic infection in the host. Host cell survives and these parasites can live longer inside the host. So depending upon the same protein, there may be slight variation, and that leads to different type of parasite strains, because they are, some of them are able to manipulate the host pathway, other are not able to manipulate the pathway. So next I will tell you how these effector molecules target the host cell death. So again I take the example of plasmodium, liver stage parasite, which means the plasmodium which infect in the hepatocytes, and these proteins can regulate the apoptosis. I will tell you in 2 slides how they regulate the apoptosis. So early during their growth, so parasite cycle in the mouse is of 48 hours in vivo and 72 hours in vitro. So in early time of development, using the tunnel assay which is used for measuring the apoptosis, you can see a red signal in 24-hour and 48-hour in those cells which are not infected. But if you see a cell which is marked by the green signal of the parasite, there you don't see any red signal. That means the cells which are infected by the parasite, they inhibit the apoptosis during these time points, which is early growth and late growth. And these can be further confirmed by a degradation of the lamin. Lamin is a protein present in the nuclear membrane, and if there is a degradation of the lamin, that indicates the apoptosis, and if there is no degradation, that indicates there is a no apoptosis. So the infected cells, which are again marked with the green, you see the clear cut red signal which presents the intact lamin. That means these parasites are able to inhibit the apoptosis. But is this happen all the time? The answer is no. Now, if you look at these different time points, 63 hours and 75 hours, which are the time when the parasite is completely mature and ready to be released. So another method of apoptosis which is used for measuring, which is called annexin 5 labeling. So at 63 hours you don't see any annexin 5 labeling, but at 75 hours you see a clean annexin 5 labeling. What it means is that up till 73 hour parasite is actively inhibiting the apoptosis, but when it is ready to be released into the host, then it is inducing the apoptosis, or it is not blocking the apoptosis. So the parasite is having a sense when to upregulate or downregulate the apoptotic pathway for its own benefit. So in B, I have shown you the labeling of annexin 5. The same can be tested by using the tunnel assay. At 75 hours you see tunnel positive cells, but not at 63 hours. And in the extreme side you see the graph quantitating the same thing. So it is indicating that plasmodium parasite is able to initially inhibit the host apoptosis, and late in the growth it actually activates or does not block the apoptosis. Similarly, toxoplasma can also inhibit the host cell apoptosis. It can utilizes the multiple ways to inhibit the host pathways. It can inhibit the caspase 8. It can inhibit the mitochondrial based apoptosis. It can inhibit the activation of caspase 3 and 7, which are the functional caspases, and it can also activate the uh, bad and bax based apoptosis pathway, which is activated from the PKB and AKT pathway. So ultimately, the using the multiple blockage of apoptosis, it effectively controls the apoptosis of the host. Next, we talk about the targeting the host of autophagy. Malaria parasite, it selectively modifies the host autophagy. Mouse malaria Plasmodium bergii infected hepatocytes. The PB membrane is decorated with LC3, which is a microtubule associated protein, and a mammalian ortholog of ATG8. So that means it is allowing the host autophagy to be modified. The parasite protein, which is called UIS3, this interferes with the LC3 binding to the selective autophagy receptor and its fusion with the lysosomes. Okay, if there is no labeling with the LC3, then there will be no fusion with the lysosome. So UIS3 in it is interfering with the LC3 binding to the its a receptor. This process is helpful in the parasite growth and survival. Another protozoan parasite called Thylaria. This also does the similar thing. LC3 LC3 is also present around the infected cells and parasite. In the green, you see the staining of the Thalaria parasite, and in red, you see the LC3 staining that tells you about the autophagy related modulation. How this Thalaria parasite inhibits the autophagy? It uses the cyclic AMP PK signaling, which is upregulated by this parasite, and this CMP PKA signaling negatively regulates the autophagy. And cyclic AMP-PK mediated phosphorylation of LC3 blocks the autophagy. So what is happening is that when the cyclic AMP-PK signaling pathway is upregulated, it actually blocks the phosphorylation of LC3, which is required for its function. In the toxoplasma in permissive host, the toxoplasma activates epidermal growth factor EGFR AKT pathway to prevent the host autophagy machinery from targeting the parasite for lysosomal degradation. Toxoplasma gondii also induces significant recruitment of autophagy machinery in HeLa and other cells where genetic inactivation of autophagic function limits parasite growth, suggesting a beneficial role for the host cell autophagy in the development of the parasite. So using this EGFR AKT pathway or by inactivating the genes, it was proven that toxoplasma is modulating the host autophagy. Next, we talk about subversion of host cell organelle and how it is beneficial for the parasites. So the toxoplasma, it can subvert the host organelles and cytoskeleton and lysosome for various purposes. To survive and to ensure its division, toxoplasma must acquire nutrients, and it is why it is must, because they are auxotrophs. That means they cannot make these nutrients on their own, so they must get these nutrients from the host. That includes tryptophan, arginine, polyamines, purines, cholesterol, iron, and other essential nutrients. So they must get it from the host, and they have to somehow modify the host, that host don't sequester these essential nutrients, or they don't block the uptake of these nutrients by the pathogen or parasite. So how the toxoplasma is utilizing the host organelles for its benefit. So mitochondria, it utilizes the mitochondria and ER of the host. Association between the parasitophorus vacuole membrane and host mitochondria and ER is seen shortly after invasion and remains stable throughout the evolution of the PB. That means as long as the toxoplasma has infected inside the cell, it it associates to the mitochondria and ER of the host. This tight association between parasitophorus vacuole membrane and host mitochondria and ER provides a key source of new phospholipids. Mitochondria is a good source of phospholipid, and when they are in a close proximity to the pathogen, they can easily take those phospholipids for its own membrane and other function. So they also manipulate the cytoskeleton and lysosome. The toxoplasma gondii infection causes reorganization of the host intermediate filaments and microtubules around the parasitophorus vacuole. Host microtubule organization center MTOC is recruited from nuclear membrane to the PBM, and host expression of transferrin receptor is upregulated in toxoplasma gondii infected cells, suggesting that parasite utilizes the host endocytic pathway for iron acquisition. So here is the picture how it is doing it. So you can see the LDL or low density lipoprotein is accumulated in these invaginations or endosomes, which endosomes also later on contains the cholesterol, and this cholesterol is taken by the this structure called the host, which are basically the invagination of the microtubule by the protein, host protein, this parasite protein GRA7. GRA7 induces this kind of structure which can directly take this cholesterol laden uh, endosomes and lysosomes. Therefore, they have direct access to the host lipids. There are, you can see there are many protein from the parasite which are different rotary proteins 4, 5, 7, 8, 18, 1. Then the granule protein 3, 5, 7, 8. Then granule protein 2, 3, 4, 6, 9. Antipages GRA1, GRA3, 5, 7, 8. These are all on the surface of this parasitophorus vacuole membrane surrounding the parasite, and they have a various functions for acquiring different nutrients to the host. For example, here ROP2 is required for association of mitochondria to the PBM, and this actively helps in lipid uptake. Similarly, GRA7 is required for formation of the host, and then there are other this ROP14 which is required for making a pore so that toxic material can be exported, or small ion ions can be taken by passive diffusion. So there are these proteins like gra 2, 3, 4, 6, 7, 9. They they make this intravascular networks for uptake of the nutrients. So you can see the various protein exported by the parasite comes on the surface and have a different function in acquiring the nutrients and modulating the host cell cytoskeleton. So we take example of malaria parasite. You see this malaria parasite growing inside the hepatocyte. In the parasite is grown shown in the green, and this is a time lapse microscopy. You can see different in numbers are showing the different seconds. Okay, and when you see towards the end like 51 to 53 seconds, you can see the small bulb like structure emerging from the infected host. These bulb like structure all called merosomes. Merosomes are nothing but the membrane coming from the host, and it contains the thousands of merozoite coming from the parasite. So why they do this kind of structure formation? Because they have to travel from liver to the lung, and if they don't have the host membrane, it will be recognized by the immune system and killed. So but if you have the host membrane, it is considered as the self, and immune system does not react. Therefore the parasite is protected from the host attack. So plasmodium can also downregulate inflammatory response by heme oxygenase upregulation. In the infected hepatocyte, expression of heme oxygenase is upregulated. This leads to proportional increase in the parasite liver load. The carbon monoxide and biliverdin, which is the enzymatic product of heme oxygenase 1, if you take that, if you don't upregulate heme oxygenase, rather you just increase the carbon monoxide levels, it can also increase the parasite load. What hemoxygenase does is that it actually controls these chemokines MCP1 and MIP1 alpha. So in a wild type, you see less level of MCP1 and MIP1 alpha, but in a knockout of HMOX, which is hemoxygenase, you see more level of MCP1, MIP1 alpha. And if you have a higher level of these chemokines, it will reduce the parasite load. And in the extreme right, you see the number of inflammatory foci. In the second or third slide, I showed you the parasite which was being killed by the neutrophils. Basically, those are called the inflammatory foci. So here you see if you have this knockout of HMOX, you see more number of inflammatory foci. That means those parasites are able to kill those host is HMOX minus host is able to kill the parasite more effectively. So presence of hemoxygenase in the host and upregulation by the parasite therefore is beneficial for the parasite. In conclusion, host pathogen interaction is like an intricate dance. Sometimes the pathogen leads and other times it is the host which is in control. Ultimately, the interaction leads to a resolution favoring one or the other or to a balanced situation in which the coexist. So you can think of those three different substrains of the toxoplasma type 1, type 2, type 3. In type 3, host and parasite both coexist. In type 1, parasite is able to kill the host, and in type 2 the parasite growth is enhanced but it is not able to kill the host. So different situation can happen by presence of these effector molecules. Thank you. Here are some
Thank,
of the references that you can if you are interested you can read. Thank you.
थैंक यू वेरी मच सर, थैंक यू वेरी मच। बहुत अच्छा से, बहुत स्मूथली, शुरू में थोड़ा टेक्निकल प्रॉब्लम हुआ, बहुत बहुत अच्छा से आपने सब एस्पेक्ट को इसमें टच किया है, बहुत अच्छा है। Now I request attendees to ask question if you have any question.